but I'm very happy to welcome Zay Bednash. Take the floor, sir. Thank you so much.
Thank you so much, Annie, for this such a sweet introduction. I'm really excited to share y'all this project from year one from Fundus. I think the beginning of the year is a perfect time to kind of recall the successes of the years and really just kind of take inventory and plan for what's going to come in the rest of the year. So CA FunDIS was a project that was started by Fundus, which is the Fungal Diversity Survey. It is a nonprofit organization that's engaging community scientists, land protectors, things like that to focus on fungi conservation and really highlight that fungi is its own kingdom in this very important kingdom that creates this interdisciplinary science around it. Fundus envisions a world one day where all the entire fungal kingdom will be fully documented, understood, appreciated, and protected. Fundus is a group of people protecting fungi. And CA FunDIS grew out of this organization and shared interests with the California Institute for Biodiversity. They truly wanted to create, as Annie said earlier, this voucher-based DNA-powered inventory that would document things that hadn't been documented, really fill gaps in historically biased areas and regions and things like that. And then really create a new data set that conservationists could then use to and point to explain why regions are very important. This was really the long-term goal. So CA FunDIS, as of right now, is going to be a 5-year project. And the conservation is really the long-term goal. But the short-term goal, starting in 2023, last year, was to really establish a high-quality field collection protocol, and that this protocol would be implemented time and time again by the team of collectors to the point where it was fine-tuned, very solid, could be shared with other people, could be replicated, and ensure that the system is becoming consistent within the organizations that are doing it. We wanted to hire collectors that would also implement these protocols. Same thing with sequence validators and sequencing, that we would build these very beautiful, intricate protocols that we could continually use, improve, and ensure that we're getting consistent results from. The other great thing of the short-term goal, which was achieved instantly, was that we partnered with California State University, East Bay, which used to be California State University, Hayward. And I was going to say Dr. Brian Perry, he's going to be the head of our herbarium. They are committed to ensuring that all the fungi that are going into this herbarium are going to be publicly accessible, so that if other people want to do microscopy on these sequences, if other people want to take them out and potentially do LSU, TEF1, other gene regions, we'll talk about that in a minute. And just to be able to make sure that this stuff isn't just private, it's not esoteric, it's not hidden behind a door. This is accessible information and accessible samples that will drive future conservations and future research and future study in fungi. CA FunDIS was going to have the 3 steps to ensure that we are creating this museum quality collections. And it's going to be the physical observation, is going out in the field, observing a mushroom and making very high quality documentation. Sequencing, which is going to be the physical sequencing of the mushroom itself, and also the validation. And then the accessioning, which comes after our sequence lead passes off the dried samples to our herbarium director and Dr. Brian Perry. But again, the first step is collection. And Fundus, I truly believe, is setting the gold standard for how to take very, very compelling documentation of fungi. You could see here in this top photo, turn my little laser pointer on. This top photo right here it's just one photo, but this one photo tells a lot. We see varying growths of mushrooms. We see varying features like the rings, the gills, the base,
I'm not sure.
things like
Sorry to
that.
interrupt. Say, I don't know if we can see your slide advancing. It may just be me. But I'm still seeing your
I
first
think
slide.
it's just you, Annie. I'm seeing the one that.
Oh, great! It's probably me. Okay, sorry. Sorry about that.
No problem. It's okay. Sorry, Annie.
No, I'm turning off my
There's some
video.
light bluing.
It's probably my dumb computer. Okay, I had to turn my video
No
off so
problem.
I don't slow down my thing. Sorry about that.
You're good. Yeah, as I was saying, just really noting the features of the fungi, but the big thing that really I think is the big takeaway from this project is going to be this Hayward session tag. And Hay stands for Hayward, which was the previous name of Cal University, East Bay. And this tag has a QR code which we can reference for the iNaturalist observations as well, that we can refer to. It gets vouchered, it gets an iNat tag as well. Yeah. Oh, I'm missing a slide. So for example, we're going to look at an example collection. So this was Stu Pickell. He found this is a Pyrenophora semi-neperta. This was actually the first ever observation on iNaturalist. It's kind of like a generalist plant pathogen of different grasses. And so if we were walking through this, Stu comes upon this mushroom and finds it in the field. He takes a very beautiful, well-documented photo of it. Excuse me. And this is going to be like the first step of the collection process. The metadata is really going to be important as well, and not just being able to see where the mushroom is and have very accurate, high quality pictures of the mushrooms, but having the overstory species and the microhabitat and descriptions of the area to make sure that not only is the area being described, but this exact spot where this fungi, this specific fruiting body was found, could be returned to again. Because if you can't find and can't come back to the exact spot that has a rare fungus, has an important sequence, has an important fungi, you cannot protect that area. So these are another great, couple great observations as well. And I think I really want to give credit to Mandy here for this incredible. I really appreciate the way Mandy observes her mushrooms. You could really get a feel for the environment here. So if you were to walk by this tree specifically, you can come back to it again in the future. There's also the Hayward tag with a pocket Garmin that is showing, I believe, 5 to 6 decimal points in the location. So you'll definitely be able to get back to the exact spot that this mushroom was growing again. We have a KOH reaction down the bottom. We have UVB here. I think it's a 265 nanometer wavelength. And again, Mandy goes above and beyond, in my opinion, with the metadata and descriptions and whatnot as well. Because another thing that we want to do in the future is, once we've compiled a lot of these sequences, compiled a lot of this data, is to be able to compare habitats and see how the species or the DNA sequences of certain species in one habitat compares with another, and whether or not fungi are going to be species dependent, whether or not they're just locational dependent and they can share different microhabitats, things like that. I'd also just like to show Michelle one of Michelle's collections as well. This is actually was a sequence we found that did not have a very close relative, so we gave it a provisional name. We will discuss how we come up with those provisional names very shortly. But again, you can note even just a few pictures here, this says a lot. We could see juniper berries, we see juniper tree, we see the difference between the fruiting body when it was here and when it was excavated by Michelle is that there is much more going on with this fungus than you'd see at the surface. Again, I like to just highlight all the collectors, not just the few that I was raving about, but Christian Schwarz is our collections lead. We have 15 collectors on the team, but there are 19 people making observations for the collections as a whole. I'm just, if you really take the time to go through each person's iNaturalist from this project, you really get to see their personalities, and the way they collect really shows people's personalities as well. I'd like to show a quick highlight to a few other folks like Damon Ty, who's been collecting like crazy for us, even though he's not fully a part of the project. Our director, Gabriella, who has just been, very helpful to me and just very empowering. Makes us feel heard, makes us feel validated. The same with Nicole and just the workplace environment that they've created in the last year. Mandy too. Mandy's just been going crazy, giving us visibility and just the contributions that Mandy had in the final report are great as well. And same with Laura Morgan as well. She's the you guys do go through the project, you'll notice any of the visual aids and any of the diagrams and stuff like that were contributed by Laura. But again, so if you want to take a peek at the project, it's just going to be inaturalist.org/projects/CA-FunDIS. And this is where you'll be able to go on and check all the observations we've made, the observations that we will have in the future. So we're currently at 50, almost 6,000 observations total for the project, because it's always ongoing. This isn't just year one, this is year 1 and 2. But you can see some of the folks that are making some of the big numbers: Dean, Mandy, Stu, Sadie, and Damon. And we can just take a peek at some of the commonly observed species. So these are going to be something that we're not going to focus on next season. We're really going to try to focus on different locations that we haven't been to and some of the species that we haven't observed as much. This is a beautiful curve or not a curve, but a graph that's showing as the CA FunDIS project really took off and started hiring more folks. Well, many observations we amassed between November of 2022 and July of 23. It's just it really goes to show how effective and how quickly a team could build and make quality observations when the funding is there, when the support is there, when your team is really working together and your people are making sure that the folks are being heard. I got a little off tangent there, but after we'll still be going off of Michelle's or Stu's collection. After they bring the mushrooms back from the field, they're going to put them in a dehydrator like this. Smaller mushrooms get, actually not sure what they're called. They're little like glass petri dishes or plastic petri dishes. We place them in the dehydrator. This just keeps everything together. Things don't move around. We don't lose track of stuff. Typical 100 to 115, 12 to 24 hours, making sure they're cracker dry. And then they're going to be placed in a plastic bag with silica just to keep any more water out. They're going to sample these after they're dry. This is quite an intricate process, and I really have to commend the team for how successful we've been and how little of contamination issues and cross-contamination issues we've had. Every time they're sending a plate off, they are doing 95 samples. So maybe not every time, but generally speaking, for ease, doing a full plate at 95 is most effective. It's very similar to a typical extraction protocol where we're just going to use a pair of forceps to pull a piece of the fungal tissue off. It's really going to be like hymenium tissue that works the best. Gills, pores work well, galebia, apothecia for like ascomycetes as well. There's a lot of care to make sure that they're not damaging the DNA, but extracting it properly. Yeah, placing them in the tubes here with forceps, and then making sure that they're not being cross-contaminated. Yeah, so after the sequencing process, or the samples are pulled apart for sampling, they are sent in this plate to our sequencing lead, Hartsinger. And originally, these were being done by Bold. I'll talk about this shortly. And we kind of graduated to Oxford Nanopore and doing massive parallel sequencing to be able to increase our throughput. I just want to highlight my team real quick. Hart has been a commending lead. Sigrid, I have to just thank Sigrid so much for everything that I've learned. Just a very, very important person who's kind of made me the person I am today. Sharon, Scott, Valerie, Justin, very, very, very important, very knowledgeable people as well. Winnie and Wolfgang are no longer a part of the team, but they helped us validate a heap of sequences too. But after the I guess we need to understand DNA a little bit before I just start talking about how we sequence. So DNA is just it's combinations of base pairs that they're nucleotide base pairs. These base pairs are going to have nitrogen in them, and they pair, I guess, just based on its chemical composition and the elements there. But thymine and cytosine pair together, guanine and adenine pair together. These are our pyrimidines and our purines. And the fact that our DNA can form these base pairs is how but how organisms replicate. That's how they produce their genetic codes to, do all of their living functions. So after we sequenced, or after the mushrooms were sampled, and sequenced, we're going to get kind of this data visualization like this. This is how we were doing our sequences to begin with. We were doing Sanger sequencing. And this is probably the sequencing form that most people are familiar with. It's where a mushroom is produced with not a mushroom, but you get a forward and reverse read based on the primers that you're using when you sequence your fungi. And together, these two reads can be, I guess, like superimposed upon each other and they generate a consensus. So you compare the like base pairs to each other. Ones that are like generates a consensus. Ones that don't create ambiguities and creates issues in your sequence. So yeah, this is how we were starting. But it's often a very difficult, in my opinion, more of a difficult sequencing to kind of analyze. You get very basic information. We're only talking about 2 reads here. And you could see in this situation where the forward did not do very well, and we had a lot of failures. We're kind of, I guess, you're kind of not a victim, but we're stuck in a position where we're relying predominantly only on 1 read as opposed to 2. You could see some generator consensus, but a lot of failures, and we are really just hopeful that the reverse is a good sequence. So what this meant is, I guess I'm getting ahead of myself, but you can only, again, you can only have your data really presented to you in one form and also analyze as well, which is the traditional NCBI blasting your information. So yeah, this limitation really created a need for us to create something better and do something in-house that was more profound. And this was our graduation to Oxford Nanopore Technologies and doing massive parallel sequencing. We had, I think it was almost 800 specimens that were compared, that were Sanger sequenced and analyzed, as well as done by Hart, our sequencing lead here, with his personal MinION software, or hardware and software. And we made the crossover instantaneously. We had very, very high accuracy in all the sequences we compared to, so much so that we were fully confident to make this our predominant means of sequencing. And it was good that we did this as well. It was about $11 to $12 per sequence when we were sending off for Bold. And we were actually able to get this down to about $3 per sequence, doing it in-house with our Oxford Nanopore technology. And also just like drastically reducing the amount of time that it took to get these sequences back, where we were waiting anywhere between 6 to like 12 weeks for some sequences to get back when we would send them to Bold, because there would be minor hiccups. We'd have customs issues, consensus generating issues, whatnot. But once we converted to doing ONT in-house, Oxford Nanopore, we eliminated all these hiccups. We could have a mushroom sequenced, validated, and published in less than a week. And one great thing that's Oxford is new and it's constantly evolving, but the great thing about this evolution of Oxford, and that we're kind of living this evolution, is that samples could be frozen, and we can, excuse me, could either use these frozen samples to resequence if we have a failure, or we can also preserve them to go and read different gene regions as well in the future. Oxford Nanopore is going to give us a little bit of a different visualization than we're used to when we produce sequences. This isn't going to be the same as Sanger sequencing, where it's fluorescent dyes that match nucleotides being passed through the Sanger sequencer. This is it's read in groups of 3 base pairs. It's either 3 or 4, and what Nanopore relies on is reading these 4 base pairs in grouping, 3 or 4 base pairs in groupings, but reading them over and over and over again. So as opposed to getting just 2 reads from a forward and reverse on a Sanger, in this point we are getting hundreds and hundreds and hundreds of reads, where Nanopore is taking all of those reads, analyzing them, and then generating a similar consensus as we're used to in Sanger. But one of the beautiful things that doesn't exist in Sanger sequencing is the fact that we get all of the reads in Nanopore. So now is something where we would have only received a forward and reverse read has now compiled for me this specific sequence that I'm looking at 66 reads. So we are comparing, I wouldn't say to a greater accuracy, but we are comparing more reads, which can highlight one of Nanopore's deficiencies, which we'll talk about shortly. But this being able to see these individual reads really highlight the ambiguities that exist in fungi, and not fungi, but the specimens that we're sequencing. And these ambiguities aren't necessarily all true or real. They could and often are errors of sequencing and errors of the base calling software by Nanopore. But from time to time, you do notice that specific ambiguities ring true throughout all consensuses and all sequences, or all the sequences that are in consensus, rather, excuse me. And we begin to take note of these polymorphisms and differences in sequences. Yeah, so we build a consensus a little differently from those 66 reads. It's in this case, this was 133 reads in consensus, so it's a similar thing. But we're able to then use MimicMap, which can really unlock a lot of the potentials with Nanopore as well. So we have the MimicMap blast, as I just said, which gives you 2 different, I guess not 2 different, but it gives you multiple different options. So you have the traditional NCBI blast, which is what we're used to, running it through NCBI. A little bit of a different format, but Nanopore, the one thing that's great about MimicMap is Stephen Russell has had it optimized to work with Nanopore. So we are able to kind of create these like adjusted scores, which are based on similar reads of the same species compared throughout the platform. So if in this case, this used to be Trapia, but it's a Fallow-Alligaster. It's taking all the reads of these species of this specific, or I guess genre as well, and comparing nucleotides, ambiguities, and seeing which ones are similar, which ones aren't. And then if they are ring true across all sequences, we can create a more accurate or not an accurate, but an adjusted score based on being able to see that these are prevalent, I guess, ambiguities and whatnot. But the cool thing about MimicMap is I call it double the trouble. So there's this MimicMap blast, and then the MimicBlast is going to be a combination of iNaturalist. I'm blanking on the the barcode of life, bold, unite databases. So a lot of the DNA powered inventories that already exist are able to blast your sequences to a lot of these unnamed and undescribed species. So this was the fallow algaster that we were speaking about. This is kind of what the validation looks like after we've analyzed it in Nanopore and have like our consensus, excuse me. We'll go ahead and post that consensus sequence to iNaturalist, and then this will now be in record for any other time people sequence fallow algaster. Through MimicMap, you'll be able to access this specific sequence. But we show that there was 130 reads in consensus, which meant our MimicMap reads, as I showed previously, this little guy here, there were more than 130 of these reads, and 130 of them were the same. This is how Nanopore builds its validation. This is how Nanopore builds its accuracy. We have the trace files so that you can build the consensus yourself if you don't agree with the information that's presented, and a MimicMap blast results, which shows you all the data we just looked at. We give it an ID. We show some of the sequences that it relates to and link them, just so if folks want to compare the sequence, if the sequence comes back something notable that somebody notices in the future, you can compare it to some other physical observations that exist on iNaturalist. Yeah, and I guess one of the big discussions that seem to come in this work is how these provisional names come from. If you're familiar with folks like Danny Miller, Rob Tullis, even like Stephen Russell, you begin to notice a lot of these names in the sequencing publication, not necessarily publications, but anywhere they are presenting the data that's coming from. And it seems to create a lot of debate, and it creates a lot of discrepancies that I think I'd like to just go over really quickly, because I don't think it's as difficult as people like to make it seem. So let's take this, for example. This was an observation Stu made in January of an Entoloma in Temecula, California. And we sequenced it. It didn't have any matches. It's close. We had a type sequence here. This is of an Entoloma aurantiophyllum in Pinium at about 96 to 98%. So this is your top hits here are not very similar to this fungus. Again, we'll take these two out because this is this sequence. But again, if you run it through all the Stephen Russell's database, the mushrooms that he's sequenced, mushrooms that Danny Miller has sequenced, Unite, Bold, things like that, we're not getting very many matches. So due to the fact that there is no matches that match this mushroom, we are confident enough to give this a provisional name. We are not saying that this is, and in this case, this Entoloma species went to Entoloma species CA 13, standing for California's 13th Entoloma. We're not claiming that this is a new mushroom, that this is a mushroom that has never been described. What this is saying is that there is no data point for this mushroom that is described. And I think one of the due diligence that's missing from a lot of this work is that these mushrooms have been described. There have been people who have done work in these states 100 years ago. And a lot of these mushrooms do have names, but just don't have sequence data. And there really is coming a point where the folks who do taxonomy need to work, it's no one's fault, but taxonomists and sequencers need to work together, because we're creating a lot of discrepancies that I don't think need to exist. And we're really just trying to understand this kingdom that we all know and love. So again, with provisional names, this is kind of a highlight of how they looked in our distributions for sequencing. On the left, we're going to be looking at, this is Ascomycetes, and to the right, we're going to have Basidiomycetes. This is the top nine orders that we've sequenced by CA FunDIS, the CA FunDIS team, showing a comparison between sequences that had matches, like known sequences, and then sequences up to, in light green are going to be ones that have had no matches. If we look at like Helitales, Pizzazales, we had a lot of new sequences, and some of that little cups and discs and whatnot that have been coming out of California this year are not only incredibly cool to look at, but are incredibly interesting as sequences too. Yeah, it's about half of our major ASCOs. Seems like we were discovering new sequences that had no points of reference, and that gap seemed a little bit smaller in basidiomycetes. Where you could see that we sequenced a lot more polypores and got a lot less new sequences. But yeah, and it's just kind of a very cool distribution to see. I think Entoloma was also left. Agaricales was left out of the basidios here I see now, because a lot of our distribution ended up being Entolomas and Tricholomas and the CHEG, the Clavariaceae, Hygrophораceae, Entolomataceae, and Geoglossiaceae. And they're really the big fungi that interact together in a lot of the California environments. Again, so Entoloma took up the majority of our sequences at about 4.5%. We totaled almost 5,000 sequences attempted at of 3,821 total species. So there were some failures, some ones we tried again. And of those attempts, we ended up publishing 2003 to iNaturalist. This was really before our cutoff for the year. This doesn't mean that we aren't, but we're finished by any means. We still have plenty more sequences to analyze. We're kind of just in a little bit of a waiting period so we could do that. But I think it's really important here to note the distribution of new sequences. If you notice the picture that I put up in the beginning on my title slide, this shows on the left is iNaturalist percent growth and MycoPortal percent growth based on the counties of California. And if should have tried to like superimpose them upon each other, but the places where we have some of the highest, 50% plus of the total sequences from these counties coming out are new sequences, are also the places that to me, generally speaking, line up with these counties that had the highest percentage of growth rate on the platforms as well. A lot of them really were, if we look here, we're talking the mountains of Los Angeles. Actually not sure what that is, maybe like the Pasadena Mountains, but very, very interesting distributions to see that some counties around like San Jose, San Francisco, some of the mountains in MODOC are really not only increasing their visibility as places that have very interesting fungi, but are also increasing their visibility of places that are generating sequences that we have not seen before. A few of the challenges that I really just want to highlight real quick is that Oxford Nanopore sounds great. It's, increases throughput drastically, where we can sequence almost, you could sequence 960 mushrooms at a clip. It's not as accurate as Sanger. So ONT is only about 99% accurate, where Sanger is 99.95. Doesn't sound like that much of a difference, but if we're talking 1,000 base pairs, like if you're reading 1,000 individual base pairs, about every 1,000 reads, you're making a mistake with Sanger, which is about a mushroom and a half, give or take. And ONT is making about 10 mistakes per 1,000 reads, which becomes much more significant as, your need for more accurate information increases. There's also a lot of bottlenecks that I've noticed with validations. We can have some site and server issues that really kind of slow things down and take up a lot of the time. And then just time in general that it takes to sequence it. because of these sequences aren't new and we haven't seen them before, they take a little bit longer. They're taking a little bit more time to sequence, to analyze, because we have to kind of dig to point references to these guys. But it's kind of the fun, it's the beauty, it's really increases our individual knowledge and ability to analyze individual sequences. And again, I guess I said earlier that due diligence that needs to be done between linking current sequences and the data that's being produced by sequencing with observations that exist by people like Shoutouts, like people like Peck, Mary Elizabeth Banning, even people that are doing much more modern mycology, like Danny Miller and Rob Tillis, and people that are still working today. But these challenges are definitely not going to stop year 2 by any means. So that distribution map I showed earlier, all those blue spots were actually 14 counties that we missed in year 1. And we make sure that we're not just going to go to all those 14 counties, but we want 50 vouchers, 50 DNA sequences from each one of those counties that we missed in year 1. This is supposed to be a project encompassing all of the fungi of California. You can't encompass all the fungi if you don't visit all the counties. There are also 10 scarce and threatened habitats that we really want to focus on. It's going to be predominantly based on, again, areas that are underserved, underreported. There are a lot of urban areas, botanical gardens, things like that, deserts, like the chaparral and the steppe have shown, in my opinion, some of the most profound sequences and some of the just beautiful mushrooms growing in sand underneath a bush in the middle of just a desert. And really focusing on like the California oak species, black oak, Engelmann oak, and really creating these group expeditions to fill these remote areas as well. Because it takes a team. You can't just hope to get a good distribution of fungi if only one person is visiting an area. If you send more people, send a larger team, you could take more area, you can cover up more space, fill more data, both literally and figuratively. And then a team's goal that it seemed overarching to me is that the team really wants to see directed conservation. And this is just going to be that our work means something, and that we've already discovered some of these sequences that seem very noteworthy and seem very kind of like a unicorn, very important, like just very, very interesting sequences. And it's going to be on resequencing these areas, resequencing these genres of fungi and looking for them in other places to see if see how substantial those individual fungi are. And when we do find that they are substantial, that we are making substantial efforts to protect that land, and work with stewards, work with organizations and things like that that are really going to drive restoration and drive conservation. Again, I don't know if Annie threw the link in the chat already, but I can drop it in a second.
I think it should be in there. Yeah, I think yeah.
Cool. Yeah, if y'all want to go and check that out and follow the project. Yeah, and iNaturalist as well. We are constantly updating information, putting new sequences in, putting new observations in. Thank you all very much for allowing me to speak. And
that was awesome.
I urge folks, I'm sorry, I just urge folks this year to really be vigilant in the world around them, speak up, engage. And I mean, we saw how I love not to just go on about the Norwood Arboricultural Project, but I love Adele. I love the work that Adele and Annika have been doing. It's been so great for me to be a part of that work. But we can see what we can accomplish when our communities work together, and when we have focused efforts on things that bring us together, like fungi and the environments that create and sustain fungi. We can understand just how important our communities are and how much power we have in our hands. So yeah, please speak up, be the change you want to see in the world. Thank you.
Thank you so much, Zay. That was awesome. I'm throwing the link one more time in the chat. It's also further up, but here it is again, just in case anyone didn't see it. And that was just awesome. I think, first of all, I love that. Thank y'all. That's awesome. It was so cool to just see even like just the incredible team that you all have created, and to hear the way that you talk about your teammates. It's like the incredible respect and learning, and you know, just being part of something like that is very special. So it's really, really awesome to hear you kind of share, and then to see the actual results. I mean, that desert stalk puff, what is it called? The Batteria species. That is so cool. You're muted right now if you're going back to it, but it is so cool. And I agree with you. I'm really into fungi that are like bold, hanging out on the edges of kind of like habitable environments, like sandy, like dry environments. And it's so amazing to see. Wow, yeah. I don't know exactly if I said it right, but Bacteria or Bacteria. Anyway, that is so cool. That is really, really cool. I can't hear you. Oh, there you go. Oh, good. Yeah, no, you probably know. You probably know. That's so awesome. That's definitely bucket list material right there. Okay, well, you definitely have some questions.
I'm sorry, did anyone have any questions?
Yeah, you do have questions. Yeah, you have
totally
questions.
Just skipped over that. I was in the slide.
No, it's all good. I'll tell you a couple of them, and then other people can jump in. So the first question was from Elizabeth Axley, and she was basically asking for someone who uses the Fundus iNaturalist project, not just the California Fundus, but the general Fundus iNaturalist project. She said, am I doing it wrong if I don't fill out the fields like associated canopy tree species and whatnot? I always try to fill out the metadata fields, but I often forget.
Am I still muted? I don't think I am. I can't see the Zoom. I'm struggling a little bit.
Oh,
but if y'all can hear me, no,
yeah, we can hear you.
Not making any mistakes. It's definitely more for the this specifically. I was implying the quality that the team, that the CA FunDIS team that has been compiled to collect these fungus for this specific project. That there's this standard that's kind of, I guess it's not expected. This is the expected standard for collectors, but no, it's really more that I wasn't trying to show this as like a belittlement that you aren't doing it as well. It's more of to me, I'm really inspired by the amount of work that a lot of people put into fungi, and especially when I'm not really feeling like I want to go out and enjoy the woods around me. Just seeing how Cisa would put the care to mention 12 different species. It's like, wow, there were 12 trees. Just it's like I got to pay a little bit more attention the next time I go out. And
well, that's so true.
For me personally,
I totally agree, and it makes me want to hunt in places, look for mushrooms in places like Norwood Park where the trees are labeled, because I honestly often don't know what the trees are. I do my best to ID them, but it can be hard. Try to take pictures of them, but it can still be hard. But it's like so cool to be in a place where you actually have a catalog and you can know exactly what the host is. So that's cool. Yeah, totally. And so someone else was asking.
Sorry, Annie, do you mind if I jump in?
Please.
Oh, thank you. This is Elizabeth Axley. Yeah, no. First of all, Zay, this is an incredible talk. Thank you so much. Also, I just want to clarify, I definitely did not take it as any sort of belittlement, not at all. It was so cool that what you were sharing. I guess I just want to make sure that I'm providing the best data. And perhaps I missed this. This is sort of a secondary question, but so with the 19 observers that are those all positions, or are any of those citizen scientists within the California Fundus?
So yeah. I guess I can just go back to the So the 15 that I showed when I highlighted the collectors, they are like paid team members that are, well, actually no. Most of the folks are paid team members. Damon is one of our volunteer collectors. I believe he's still volunteering to collect. But yeah, no, this has essentially been a team that because of it being a partnership with the Cal State and the state of California, you know, providing funding for this, that we're able to, you know, build the team of specific collectors. 19 is so this is the 15 collectors from the team, plus Damon, plus an observation from Gabriella, plus observations from Ken. So that, I think that kind of gets it up to the 19.
Incredible. Yeah. Do you see any
19,
like,
yeah.
do you think that this sort of thing could happen beyond California? Like I'm thinking DC, Maryland, Virginia, you know, our area. Obviously, I don't know if there would necessarily be the funding for those same positions, but if they could, like, even just citizen science based.
Oh, for sure. Yeah, it's kind of a little bit of like a good opportunity mixed with it was really just kind of like a low-hanging opportunity that I think that there's so many of the people on the team are just interdisciplinary education, like people who educate others, they're plant people, they're fungi people. I mean, Damon's like he focuses on California sturgeon. So we have quite an immense background of folks. And I think that really, that's what I was talking about, like the personality, is that some folks care about plant pathogens and will only take pictures of something that you cannot see with your naked eye, something that you can only see with a macroscope or a hand lens. And then some folks are only looking for polypores because they're convinced that, you know, the polypores have the best distribution, and they often find people are like, oh, there's nothing left, we found them all, and then they'll bring back 6 new sequences. So it's kind of, it was not to tangent again, but I think that the fact that so many of these folks were already focused in California on so many different topics really created this perfect opportunity. But it's like, I mean, we know that MAW has such, for example, MAW has so many different members of interdisciplinary facets and different fields of education and research and things that they'd like to pursue. So I think it would be very doable to, you know, be able to pick it up and implement it in other places.
Awesome. Well, that was a great
question.
Yeah, that was a great question. Another question in the chat was about you know, you mentioned that taxonomists may have you know, their own collections or may have their collections in different herbaria. And I think maybe it was Elizabeth and Matt were asking, like, would the USDA herbarium be one where there might be collections of species that have been named but that haven't been sequenced?
Oh, most definitely. I think that's I totally forgot to leave that out of the goals for year 2, is that we want to sequence mushrooms in other herbariums, that we want to be able to start to draw comparisons to the things that we've sequenced, to see you know, all the new sequences that I mentioned, to see how novel they are, to see if they truly are new sequences or if they have matches to things that haven't been sequenced. I'm not sure which herbariums we want to sequence, but I believe we're working with UCLA to do UCLA's herbarium. But I again, I can't speak on that. No.
But that's awesome.
Sure.
That's really cool. Like, I feel like, because I loved your slide with the provisional names, and I think you're right. Like, it does cause a lot of conniptions among people to, you know, see these names thrown out. And I think your explanation was really beautiful about how the names are like a way to communicate about something. It's not necessarily saying this is new, and it's not necessarily saying this is a species, but it's saying, like, here's how we can talk about, you know, this thing. And
yeah,
and there's so much
it's
like,
something.
yeah. And it's begging for people to do that work, connecting it to the data that may be hiding somewhere in an herbarium. So that's really, really, really cool. If anyone wants to jump on and ask additional questions, you you're getting a lot of excellence and awesomes and thank yous and wows. And so that I agree. I think that was terrific. But this is a great opportunity for folks to, unmute yourself and ask your questions to Zay. And I wanted to just say, I think it's incredible that, we have members who are doing this kind of work, and what Zay is working on with this team, not alone, but with this team, is like such an example of something that we could do here. Fundus is funding regional projects. So if someone wanted to make a regional project, they could make one for our region. And, MAWDC, obviously, that our DNA committee is doing a ton of amazing sequencing work too. So I would just think anyone should feel empowered that, like if we look to what our members are already doing, we can see a path forward for coming up with some really special conservation projects. Anybody want to hop on? I see something, let's see, popping up in the chat. Oh, Megan said MycoPortal is a global clearinghouse for fungal collections and would be where you would go to find type or other voucher collection information. That's cool, MycoPortal. And Zay, you referenced MycoPortal, I think, too. Yeah.
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 I believe MimicMap is starting to pull collections from MycoPortal too. I'm not entirely sure about that, but it's definitely a very, very good resource.
Awesome. And Unite, you said, I think, as well, which is like another. Yes. Cool.
Yeah. And Unite can be a little difficult to navigate, but I think it can have quite a heap of European, especially for European taxa, like European like sequenced fungi.
Well, if anyone wants to ask a question, ask away. And otherwise, thank you so much to Adele and to Zay and to April and to Mitch and Elizabeth. And I feel like there's so many amazing people all around us. We're so lucky to have each other, and really excited to start the new year. We're going to have our next meeting IRL. That means in person. So it'll be a really great chance for us to all connect, and really we can put our heads together for what we want to do. I mean, we have a huge club and we have a number of things going on, a number of different subgroups. And I think there's a lot of opportunity for kind of new ideas and synergy here. So I hope this was inspiring to you. It was extremely inspiring to me. Elizabeth, you said what? IRL? That's right. The Britt Bunyard talk on February 11th, I think. Sunday, February 11th, is IRL, and that's going to be our meeting. So please come to it. It's at the Mount Rainier Nature Center, just as a reminder, and that's where our incredible secretary and board member, Serenella Linares, has been doing amazing work there since I guess this was her first year there as the naturalist for that park. So anyway, I think that'll be an amazing event. I hope to see all of you there, and really, like, we should start these conversations. So thank you, Zay and Adele, for the inspiration, and everybody else, and Happy New Year.
Yes, thanks everybody. And don't forget also to check out Tom and Natalie's fungal and lichen walk this weekend at Great Falls. So that info is up on our website as well. All right. Good night, everybody. Take care.
Good night.